So my name is Kris. I head the CBD Bioinformatics single cell bioinformatics expertise unit. And I primarily do service for the labs in the department, but I also do some development work for a few different tools that I use with the lab, the department, and kind of the wider bioinformatics community. So I'm going to go over two of those tools today and hopefully give an end-to-end demo during the talk, and you'll kind of see what I mean as I go. And those two tools are VSN and SCope. And so they are both used for analysis and visualization of single cell data. So a very brief preamble on single cell data. It is typically very large and complicated to analyze. There are lots of different tools. There are lots of different ways to analyze single cell data. And these kind of things can really be a barrier for use, both academically and in industry. Just because for anyone that wants to use this data, it looks very unwieldy and very hard to get into. And on top of that, there is a lot of data being generated or has been generated. There are a lot of atlases, for example, the Human Cell Atlas, the Fly Cell Atlas, and the Tabula Muris project, which are generating hundreds of thousands, if not millions, of single cells across every organ of different organisms. And these are now reaching not only into kind of wild type, but also into conditions, treatment, into diseases or different conditions of those organisms or tissues. And that amount of data has a large use in a lot of different projects. A lot of people may not realize that that's available for them, or again, they may know that that data is available, but it's not analyzed in a particular way that's helpful to them, and they want to go further and they want to look into it in a different way. And although a lot of non-single cell data have pipelines for ease of use and reproducibility, the NF-core pipelines are a great example here. And admittedly, they have an early single cell RNA-seq pipeline. Their bulk RNA-seq pipelines are very easy to use, for example. Single cell doesn't have this so much. And this is where VSN comes in. So VSN, or VIB single cell NF, which is shortened to VSN or VSN as we name it, is a next flow pipeline that is designed for the analysis of single cell RNA-seq data and eventually other modalities. It is a collection of different tools and workflows under a single common framework. And I mentioned this is powered by Nextflow. And this is a collection of tools that we've kind of pulled in, but it's open source. People can contribute. They can bring in their own tools and things. And it provides an end-to-end solution for a basic single cell analysis. It's not fully comprehensive. It can't do everything. But if you want to get a bit of insight on a set of data, then it is a really good way to do that in the first hand. It's automatable, it is reproducible, and it is scalable. This has primarily been developed by Mats and Kris from Stain's lab, myself, and also in collaboration with the lab of Ivan Seys in Ghent and Janssen Pharmaceutica. VSN isn't magic. It's not a replacement for bioinformaticians, and it doesn't immediately perform complete and full analyses ready for publication. I mentioned that in the previous slide, it's automated, it's reproducible, it's scalable. These are really useful things for publication, but it's not just a set it, forget it, and use the results as is. You really need to go through and check things still, because single cell data has a lot of nuances about it, and you need to make a lot of decisions at various points. So how can you run it? And this is the first point where we're going to work out how my demo is going to work. To show how easy it is and how quick it is to run, I'm going to give an end-to-end demo of VSN and SCope as I give the talk. So I'm going to switch to my terminal, and I'm going to show you how to run this. I can post these commands in the Zoom chat later. You can also find them very easily in our GitHub repositories. The two prerequisites you need are Nextflow installed, which is a very easy install. You can install it as a user as well. You also need some kind of container system installed, which is either Docker or Singularity. For reference, I'm running this on my local computer at home. This is not on a powerful server or anything anywhere, and it's a Windows machine. It's using WSL2 for anyone that's familiar. And I have both Nextflow and I have Singularity installed. So the first thing I'm going to do is tell Nextflow to pull our pipeline. So we pull just using the GitHub repository ID. Normally, this will then check it out. In my case, I've already got it. It's up to date, so that's fine. If it's not up to date, then it will pull the latest version of it. But it's all automated as long as the repository is public, which ours is. Nextflow will do all of the handling and everything like that for you. The next command I'm going to run, and these are all fully detailed on the read the docs page, which there's a link to, and you can find it just by Googling VIB single cell NF. This is going to generate a config file. So we tell Nextflow to generate a config for the repository. We're going to tell it which profiles we want to do. In this case, we're inputting 10x data, which is single cell data from the 10x platform. We want to run the single cell pipeline, and I'm specifying specifically that I want to use Singularity as the container system. I'm also outputting this into a config file. So take a second to run. I'm outputting it to a file, so it's just going to complete. Before I do the next step, I'm going to download the data. I'm really hoping this doesn't mess with my camera and my stream here, but I'm just going to download a 10x example dataset. So this is a dataset from 10x's V3 chemistry. It's 10K cells from PBMCs. So I'm going to pull this in. It should only take a few seconds. While this goes, I'm going to copy the next command. It's only 80 megs, so it's pretty quick. There we go. I'm going to make a new directory where I'm going to store the data, and I'm going to extract that data. And if I list this folder, you'll see that this is just a traditional 10x folder with the traditional 10x files. There's nothing special about this. It's directly from their website. So the next thing I'm going to do is I'm going to open up my text editor, and let's see if this works. There we go. And I'm going to modify the config file. So that config file command I ran generates this file. And there's a manifest at the top, which is important for the reproducibility. This specifies which version of the pipeline you used. It also locks it to a specific version of Nextflow so that you can't run it when there's not a supported version, for example. And all of the parameters used to do the entire run are stored in this config file. And this is what makes part of what makes Vision so reproducible. You've got simple things like your project name. You can rename anything like this. You've also got for each of the parts of the pipeline, in this case, we're using ScanPy for the single cell analysis. You've got all of the different individual steps, feature selection, scaling, the neighborhood graph, et cetera, and what the parameters are. So for example, filtering, what are the minimum number of genes you want to include for a cell? In this case, the default is 200. I'm going to change a couple of small things here. So I am going to first update the data. So the data is here. This is the default path. I'm just going to update this with where I just downloaded and put the data. I'm running this on my local computer, so I do not have this much memory in my computer. So I'm going to adjust that quickly. And I need to remove one parameter at the bottom here, which is a default for use on the Flemish supercomputer. That'll actually be removed in a future version. There's a lot of different things you can do in the config file. I'm not going to go over all of them now, but be aware that this is where the heart of what you're going to do is. All the parameters are stored here, and as long as you run the config command correctly, you will get everything that you need in this file. So I'm going to pop back to my terminal now. I have just saved that file, and I am going to start Nextflow running. So with that, I provide the Nextflow command. I tell it specifically I want to use the config file that I saved and I've just amended. Again, I'm running the VSN pipelines repository, and in this case, I just need to tell it the entry is the single sample pipeline. And this means that it will run the single cell pipeline, and that's what will run for Nextflow. Fingers crossed this runs perfectly, because demos are always dangerous when you're doing them live. So I'm going to give it 2 seconds to start up. You'll see it takes a second just to work out everything it's got to do. One of the things it does here is make sure that all of the parameters are correct and everything. Perfect, it started. So with this, I should now be able to do this. Perfect. We're going to leave that going up in the top right-hand corner so you can see it's going live. There's no magic here. And you'll see just how fast it runs. This is, again, for 10K cells on my local computer. So once you've done all of your config, edited your config and settings, pointing to references and everything, and you've got it running, the combination of the config that I defined and the pipeline version is fully reproducible. So one of the things that's important there is some processes are stochastic. They require an input seed, and we're pretty careful in defining this at every step. I didn't show it, but in the config file, you can define a seed, and that means that all of these random processes are locked to that seed. And so if you rerun, as long as you use the same version of the pipeline, the same config file, and the same input data, you will get the same output data, no problem. One of the other nice things about this is Nextflow has functionality built in for resuming. So if my pipeline got stuck halfway or the server crashed or something, I would be able to use the -resume option. And as long as the input files are the same, the config is the same, it will pick up where it left off. So a bit more in depth while it runs in the background, what is Vision? I mentioned before, it's a collection of tools and workflows, and the GitHub link is here. We've also made Vision citable. So we use Zenodo to get a DOI for not only Vision as a whole, as a project, but also each version of the project gets its own DOI. This is pretty cool because it means if you happen to use it in a publication, you don't just reference the tool, you reference the explicit version of the tool that you used, which means anyone can find that DOI and go back to the exact code you used to run the pipeline. And again, I mentioned it's versioned and reproducible. Each of the tools we provide, so ScanPy, et cetera, are versioned, and we provide versioned Docker containers. We write everything in Docker, but they work perfectly fine in Singularity, as you can see running in the corner. And this is, again, part of reproducibility. As versions of software updates, sometimes something updates and it changes. So by using specific versions of software, which are defined in the config file, our containers, then you have no problem there. There's one exception with SailRanger due to licensing, but at that point, we provide instructions to build your own container and use your own container. The processes and scripts are all used in workflows, but the scripts we use can actually be pulled out of the repository and used elsewhere. And we have a Read the Docs website, which is got a lot of information about running it. It's got some tutorials, some examples on doing different types of runs. So some of the different modules and pipelines we have include raw data processing. So I mentioned SailRanger, so that will do from either raw BCL files or FastQ files to run the SailRanger pipeline in various different ways, including using counting the antibody counts for site-seek or hashing, or merging multiple 10x runs together. There is a pipeline for running demuxlit or freemuxlit if you happen to have combined your samples from different patients or from different genotypes, then you can demultiplex them using this. And we have a pipeline for running the Nemesh pipeline for dropsec data as well. For the single sample, so these are where we expect it's one data set in, one data set out. We have single sample, which is the one that's running in the background, and that's a ScanPy analysis. We also have this in combination with Scenic, which is used for gene regulatory network inference. Scenic on its own, if you've already got a data set, you've done all your analysis, you just want to run Scenic. Scenic multi-runs, which is actually, Scenic is stochastic. If you run it a couple of times, you will get different results. You can provide feeds to get the same results, but so Scenic multi-runs actually allows you to run this multiple times, automated through Vision, and it will combine everything into one file at the end. And you can say, only show me regulons that showed up in 5 of the 10 runs, for example. And finally, single sample Scenic multi-runs is pretty self-explanatory. On the other hand, we also have sample aggregation pipelines. So this is where multiple samples in, one data set or one analysis out. And you can do this with just no correction, or you can do it with correction. So we've got BBKNN, Harmony, and MNN correct as ways to do batch correction on the data when you do that. So the samples come in and each sample is used as a batch key and they're corrected and the rest of the analysis goes on as is. So a couple of other features that you get by using Vision is you can have sample-specific configurations. This is pretty useful for people like me who maybe process an entire run of data, but there's different samples that need different parameters. For example, I had a run not long ago where I had 3 samples from one project, but 2 of them were from one species and another one was from a different species. Using the one Vision config, I can actually specify that one of these files needs a different transcriptome to the others for the mapping steps. And that's the only difference. Everything else will continue as is. And I was able to do that pretty easily. Again, details for all of these are on the Read the Docs website. We have automatic determination for principal component analysis, numbers to use further in the analysis. So you typically have to make a decision on how many to use for the neighborhood embedding, the t-SNE, the UMAP, etc. We have automated determination of that, and it's built in. Some parameters can go through parameter exploration. So this is an interesting one for clustering, for example. If you don't know the resolution to use, rather than run the pipeline 6 or 7 times choosing different resolutions, you can provide an array of values, 0.8, 1, 2, 4, 8, for example, and Vision will run all of those on its own, and then it will combine the results into a single file again. This is only where it's supported in specific parameters, but clustering is one example. And you can run directly from SRA or GEO data. And there's an example, a very quick overview of how you would do that here. For example, this is a cell atlas from the Drosophila midgut. You can see there's 4 samples listed on the right. 2 of these are 10x, and you can see the SRA project number showing up in red here. By adding the SRA profile when you generate the config, and by defining your data in this manner, you give a list where the ID is the SRP number and the samples can be a glob. So in this case, we just say 10x star. This is now going to take both 10x samples, and you can see in the top right-hand corner that the run is now finished. So that took about 6 minutes to do that run of 10K cells, and we'll come back to that in a minute. For the SRA, you can also have multiple SRP numbers here. So you could put a second run and have a second data set, right? You could bring in that as well. And then these require their own entry points because there's handling required from the SRA side. The outputs of Vision depend on the workflow that you're running. But for the most part, we try to output reports in the form of notebooks. These are computed during the pipeline. They're combined at the end, but we try to keep them runnable by the user if they want to, assuming they're using the correct kind of environments and they have the correct output files. And we try for as many workflows as possible, at least complete workflows, to provide Loom files for output to scope. And I will show you that included. This includes scenic results when they're there and the metadata as well. Intermediate files are always stored, although typically these are in your work directory, and this can get very large if you don't clear it out. So we typically delete these regularly. In the future for Vision, we want to add a lot of things. It requires time and development, of course, and we're very busy. But for example, some things we'd like to include are some advanced differential analysis between clusters, for example, sample subsetting. We will update to the newest Nextflow version soon, which actually is going to allow us to use command line options that we couldn't use before. NF Tower compatibility, which is a web viewer and manager for Nextflow pipelines. And there is in development and hopefully coming very soon is a Surat pipeline to go alongside the ScanPy pipeline. So although we output Loom files, we also output H5AD for the ScanPy pipeline. We would output the Surat object in an RDS file for the Surat pipeline. And that would mean that if you want to do further analysis, you can just take that RDS object and load it into Surat and then continue with whatever extra analysis you would want to do on the data set afterwards. In the long term, we have thoughts for multi-omics, spatial single cell ATAC, and things like that. So I'm going to jump to scope now. Before I do, no, I will jump to scope now, but we'll come back to the demo in 2 seconds because I will bring that Loom file that it finished and show you in scope. And I'll come back to the report that Vision has just output as well at the time. And in fact, I will tell you what, I will show the report now. That makes more sense. So I'm going to open up my web browser and my Explorer window so I can show you what the output you'll actually get is. So the outputs in terms of the reports, you're going to get a notebook that's going to look something along the lines of this. So this is our output notebook from Vision. This is the final one from the single sample pipeline. We include the whole parameters and the manifest that was included here just as reference, basically. Normally, if you open the IPyMB, this would be hidden because it's the HTML that's shown. But this report contains all of the parameters you specifically defined. The top of this is all just defining a few functions that are that are used, but then as you go down, you have all of the different graphs and everything that we generate as part of the pipeline. So we have diagnostic reports, including the highest. These are very typical ones you would usually see during single cell analysis. For example, highly expressed genes. We show you the genes expressed per cell, counts per cell, etc. And the red lines are the thresholds that were in the parameters file. If you change your thresholds in the parameters file, these would update live, and these graphs would be the values that you put instead. We show the pre-filtering and the post-filtering data, so you can see how it changed. So for example, in this example, you can see that there was a small population with few counts, few genes, a very high mitochondrial percentage, very typical for apoptotic or dying cells. And in the post-filtering, these are gone, and the scale has jumped up, which is why now everything is increased. Beyond that, we also give you the reports for highly variable gene selection, for the dimensionality reductions, the TSNE, the UMAP, the PCA, and the clustering if any was done. So here sample ID is a single color, but if it was, if you had the multiple samples, you'd be able to see each of those here as well. So this is the clustering that you then get out of it. If you were using the Leiden algorithm instead, that would be down here, or if you use both for parameter exploration, they would both be in here. So I will jump back to the presentation quickly. So scope, I've mentioned it a couple of times now. Some of you may know it, some of you may not, but what it is a viewer for single cell data. It's based on Loom files, which I mentioned are produced by Vision, with a custom metadata structure, which lets us display a lot more rich data. It's not limited to RNA. Most people do use it for RNA, but basically any matrix can be loaded. So all you require is one embedding, a t-SNE, UMAP, PCA. Doesn't have to be computed. It could be coordinates. It can be anything. One cell ID's column attribute, so we can uniquely identify cells, and one gene row attribute. And this doesn't have to be genes. It could be regions. It could be anything you want. It could be proteins for proteomics, etc. You can add annotations to clusters, and you can extract them afterwards. You can plot trajectories and things like this. So I've noted here demo on VSN output. So we will jump back to scope. And I'm going to take that file that came out of the Nextflow demo. So Nextflow demo, and this is the output Loom file. And I'm going to upload this file. This file is straight out of Vision. So this is on my local computer. It's pretty quick to upload. And in this case, it's an 80 meg file, I think, again, or 30 meg. In Scope, as a user, you get a free session you can use for 5 days. We clear data after 5 days, just so we don't end up as a repository for everyone storing all of their data, but you can happily re-upload things as and when. There is command line access if you really needed it, but we don't have that very often. So once it's uploaded, you'll see in user uploaded, we've got an unknown. I didn't specify anything in the config, so it shows here. And this is the output. So this is the output directly out of Vision as it is just run. We can do things like search for different cell types. So these are PBMCs. So we could search, for example, for IL-7R, which will show us the CD4 T cells, and they're highlighted there. In Scope, we let you show 3 different colors simultaneously. So these are red, green, and blue. They combine much like you would imagine. So for example, we can then search CD8, for example, CD8A. For the CD8 T cells. And so you can see that now there's some cells that are a bit yellow where they've got a little bit of expression of both of these. We could show also the B cells with another gene, for example. And this kind of combinatorial analysis really lets you identify specific subtypes of cells. You can see this purple group here are expressing both MS4A1 and IL-7R. The ones in gray over here, they don't express any of these genes. They are 0 values. Anything with gray is a 0. And the black are got very low expression of one of these genes. So you can show genes in Scope. What else can you do? You can also see individual clusters. So you'll notice that in this case, IL-7R is identified as a marker of a lot of these clusters. This is again built into Scope, built into Vision. But if I selected cluster 0, for example, that red color now shifts from being the gene expression of that gene to just the cells that were in that cluster. And you also get the marker genes from that cluster as defined by the marker genes function in ScanPy. You get the adjusted p-value and the average log fold change, and you can sort on these. You can go through, you can click one and it will highlight it up. If I click this one, for example, it will replace the red color. Instead of the cluster now, I'm looking at that specific gene. I will go back to that cluster just to show a few other things. You can download this table of markers. One new implementation is you can actually run gprofiler on this list. So if you were to click this link, you can select the top 100 genes sorted by, for example, log fold change, and you can say, I want to, this is human, not Homo sapiens. And you can run gprofiler. And so gprofiler is a gene ontology enrichment tool. And so you will end up with the different GO terms for those different parts. So we can see neutrophil, leukocyte degranulation, neutrophil activation, a lot of immune terms which make a lot of sense here. A few other things that I will show briefly before the end of my talk is if you wanted to, you can add annotations. So up here, you'll see it says, welcome me. I'm logged in with my org ID. You can authenticate with your org ID. And primarily all that does at the minute is gives you access to add annotations to clusters. So for example, I've used MS4A1 to identify these cells at the bottom as the B cells. One interesting thing I can do here is I can actually select these cells with the lasso tool, and I get this table on the right here. And this tells me at Louvain resolution 8, cluster 3, I have selected 100% of that cluster. Only 60% of the cells I selected are in that cluster, but 100% of that cluster has been selected. So if I take that cluster 3 and I go and look for it, you should see that if I turn off my cell selection here, indeed, this bit on the left is in red, and this has been selected as cluster 3. So I know that that is B cells now. So I can actually look for that. I can actually add that annotation. So by clicking the add annotation button that was on the right here, I can search for vocabulary. I can search for B cell, and this uses the EBI's ontology lookup service to look up for these. I can select B cell. If I wanted, I can add some extra evidence here. I'm not going to, but I could. You could also add free text if you want. So say your vocabulary wasn't there, you could type whatever you wanted here. I will add the vocabulary just because it's there. And I can submit the annotation. And now what we should see is that my term on the right shows up as an annotation, shows that I added it as a B cell. If I did provide markers or a publication of why I thought this was these cells, it would show up here, and people can endorse this. They can thumbs up, thumbs down if they agree, disagree with me. So one cool thing here then is if I now look for one other thing you can do in Scope is get an overview of your clustering. So I'm going to search for all clusters. You'll see that the labels are showing up here. And actually now I've identified this cluster down the bottom here as B cells. Because that annotation is the most voted up annotation, Scope takes it over as the name of the cluster. And so it now displays as B cells. There are a few other things you can do in Scope. The compare tab is particularly interesting if you have complex data sets. I'll give one very quick example on a public data set. So this is one of the publicly available ones. These genes don't exist in here, which is why it isn't like this. So I'm going to clear these out. Okay, perfect. And in this case, we have annotations we've added to the data. Vision by default doesn't output these. You need to know something about the cells, unless you have multi-sample, in which case your samples would be here as a batch key. But one very quick example of how powerful the compare tab can be is if I show you the male and female cells separately in two views here. So they look the same, but actually on the left, we're only showing the male cells. On the right, we're only showing the female cells. And in Drosophila, at least, there is a couple of genes that are male specific. In this case, ROX1 is them. So you can see on the left, ROX1 is expressed only in the male cells and not in the female cells. So if you had a different type of annotation here, for example, we have genotype and we have age, you could separate these out in different ways and see how your genes of interest are different across the different parts of this. So I mentioned a few of these new features, the labels, the cluster level annotation, the labels for clusters, etc. in the demo I just gave. In the future, we'll have a few other plans for Scope. We want to expand that ORCID ID login into a proper user system where you can have your own data sets, you can share them with people, etc. We're doing some refactoring on the front end, back end to improve the code and to make things a bit faster. I'm working currently on improving the viewer because when you get to around 400,000 cells, things slow down a lot. So we have an early implementation of something that should speed that up. In the longer term, we plan to support more input types, for example, H5AD for ScanPy analysis. That will be the first step. Better ATAC support and other modality support and possibly spatial support as well. And with that, hopefully you found the demo useful. I'm glad it seemed to work for the whole way through. So I'll thank everyone from the lab, Stain, Max, Kris, and Gert for all the input on Scope and Vision, and everyone else from the lab, the Ivan Safe Lab and Janssen for the Vision input. And thank you for listening. This would be where I then thank myself, and I would ask the audience if you have any questions on anything I just presented on.
Yeah, I have. First, thank you so much for Scope. I don't use the other one yet, but Scope we use a lot in my lab and people love it because it's really user-friendly. But I have a question about the, we use a lot of the compare tab as well. But I have a question about the thumbs up, thumb down, if you can add annotation. So you mean that it's already, you can share your data with other people? Because sometimes I work on projects that are multiple people are working on. You can actually share. I wasn't aware that you could share your data with some people.
Yeah, so in terms of how Scope works at the minute, the session you get is, in fact, you can't see up here because I've got it trimmed off. But let me just bring this up and you should be able to see this. Yep. So you can see the address bar. There's this UUID here, which is by default what you get for a new session. Actually, if you share that with anyone, they'll be able to see the data you see. You can, in the future, we'll allow more permanent sessions. At the minute, we're kind of limited to, we limit them to 5 days just for storage implications. But when we have a user system where we can track all the files reliably, we'll expand this. But indeed, so if I go back to cluster 3, I think I annotated. By the way, if I now search for B cell here as well, this will show up as that it was the suggested annotation. Anyone else that comes to the data set can thumbs up, thumbs down. So I can change my mind and decide, actually, I don't like that idea, and I don't like the fact that I named it as a B cell. And if I go back to all clusters here, it's back to unannotated cluster because there's no positive annotation. This can be really useful if you've got, like you said, a group of people working on one data set to annotate. And actually, this feature was driven by Scope's use in the Fly Cell Atlas. So there we have permanent sessions that don't expire after 5 days. And we've had experts from all over the world working on the same data set and annotating them with this manner. And then if you download the Loom files, all this information is stored in it and you can access it programmatically via R or Python.
Okay, perfect. Perfect. And 5 days, plenty enough actually for annotation at the beginning.
Yeah, I guess. I mean, like we said, hopefully when there's user systems, we'll be able to track Loom files and who's using what space for longer, and we'll have a better idea on how much space we're using. So then we can expand it a bit if necessary. So.
Okay. Thank you.
Yeah, no problem. Okay, I see one. Yeah, sure, Alejandro. And then I'll go to the one in chat.
It's obviously a super great effort and it helps a lot, all the tools that you developed, so the vision and the scope. So how can we help out from the outside, right? So what's the best way for external developers to increase the modularity of vision or something like that?
So both Vision and Scope are completely open source. You can find them on GitHub. Vision has a contributing document, which we plan on expanding a bit because it can be a bit daunting for new people. If there's a particular component you want, at a minimum, let us know. Open an issue on GitHub on either Scope or Vision so that we know there's a demand for something. If we don't know people want something, we're not going to develop it unless we particularly want it. Going further, feel free to, put in a pull request for some code or something. For Scope, there's Max, myself, and James from VIB Bits. And from Vision, there's Max, myself, and Kris. We all will go over any pull requests that come in, discuss them, go over with you and things. I don't think we're ever adverse to including something unless it goes outside of the vision of what we have for things, right? Like Scope is a visualization tool. We don't plan to add analysis, or at least not complicated analysis there.
I was more referring to Vision.
Yeah, so for Vision, really, if you have ideas, if you have things you would like to contribute, either get in touch with me and if it's a bigger thing, we can discuss. If it's smaller parts, like a small script or something to go along outside a module, feel free to check out how we do things, check out the contributing guide and put in a pull request on GitHub.
No, okay. Great. So there is a contributors guide somewhere in the documentation.
There is, yeah. So there is a contributing on the read the docs. There's a brief where it's listed under development, and there's a contributing guidelines, I think, in the GitHub. But importantly, if you find that something's missing there, let us know as well, and then we can build on that. We can add it, and that makes it easier for people to contribute as well.
Okay, great. Thanks.
Yeah, no problem. And then Samina asks, which options have we embedded in Scope for sample aggregation and batch vector? So in Scope, we don't do the analysis like I mentioned, but in Vision, we have BBKNN, we have MNN correct, and we have Harmony as 3 different methods you could use for sample integration. I think you can also do it without integration. So if you didn't have batch effects between your data, you just wanted to concatenate the matrices, you can do that as well. Those results again can come to Scope, and then you can see, for example, which samples you've included. Normally you would then be able to, I think I can show that here. As a proxy, I will show genotype. But if you run on Scope, run through, I can't type today, genotype. you'll see that you've got the two colors here, one for the two different genotypes we have. If you ran through Vision with one of the batch corrections, you would have sample or batch, and these would show up as the different values that came in, typically the sample names, and you'd be able to at least see where your original samples were. Okay, well, if there's nothing else, I say this always, feel free to contact me if you have any other questions or want to contribute or anything like that. I will thank everyone again for coming to the Bioinformatics and AI meeting this month. As usual, you'll receive the schedule next month for next month's speakers, and hopefully we'll see you then.